Hello everyone, in this video we will answer a few questions from Chapter 7, Mutation from the Pka Biology KML. We will answer a few selected questions such as question number 1, number 3 and number 5. First, let's take a look into question number 1. So there are three types of mutation, mutation A, mutation B and mutation C. And we will see question number 1A. Explain what is meant by point mutation. First, let's arrange the nucleotide on the DNA or mRNA into a frame that consists of three nucleotides. This will make it easier for us to trace which bases or which codon or which frame that has undergone mutation. For question number 1A, what is meant by point mutation, we will emphasize on the change in a single nucleotide pair that occur in a single gene or in a DNA sequence. Some clue for missense mutation: different codon, different amino acid, and different protein. For the nonsense mutation, different codon change into stop codon and it will produce short protein. While for the silent mutation, different codon but still code for the same amino acid and same protein. Now we will compare between mutation B and mutation C. Basically, we will compare between nonsense mutation and silent mutation. For the similarities, both will involve base substitution and both will involve single base replaced by another base. And for the differences, we will look into the effect of the nonsense and silent mutation. And then we will explain what happens to the codon and what happens to the amino acid. And then finally, we will explain what happens or what is the effect to the polypeptide. Mutation B is nonsense mutation, while mutation C is silent mutation. The codon in mutation B changes into stop codon and does not code for any amino acid. While in mutation C, the codon is changed into different codon but still code for the same amino acid. For the polypeptide, for this mutation B, the polypeptide will become short, while for the mutation C, the polypeptide remain the same. Alright, now for the question number 3A, we will look into the effect of base deletion that will lead to the frame shift mutation. And then for the question number 3B, we will apply the concept of one codon consists of triplet bases or three nucleotides, and we will see how the frame shift mutation can lead to the change in the reading frames of codons. If T is deleted, then the frame shift mutation occurs. The reading frame will shift to the left. The new sequence will be ACC, AGA, ACG, CAG, CT. The next questions explain the effect of mutational event or frame shift mutation start from the point of mutation. Started from the point of mutation, all DNA sequences change that will cause triplet bases or codons on mRNA will be read wrongly. That will code for a different amino acid that produce different or non-functional protein. For question number 3B1 and 2, the frame shift mutations occurs at 85 base. So 84 bases still correct. 84 bases equal to 28 codons or 28 amino acids. For question number 3B3, how many amino acids coded wrongly? There will be a total of 105 nucleotides. It's equal to 35 codons. Assumes that the last codon is stop codon, so the total of wrong codons are 6 codons. 35 minus 29. Question number 5 is related to the chromosomal mutation due to the non-disjunction. The non-disjunction occurs during meiosis 1 of oogenesis, produces abnormal eggs. The abnormal eggs fertilize by normal sperms to produce individual pqRNAs. Non-disjunction is the failure of homologous chromosome to separate during meiosis 1 or the failure of sister chromatid to separate during meiosis 2. The genotype of individual S is YO. It's formed when abnormal egg without X chromosome fertilized by normal sperm with Y chromosome. The zygote eggs will fail to develop or not viable. That's all for now. We wish you all the best and thank you.